Hi friends, Mr. Rogers. Today we're going to look at the cell membrane. We call this the phospholipid bilayer. Now, why it's called a phospholipid bilayer, right off the bat here, is we already learned about phospholipids. They're like a little jellyfish here. So you can see them right here, right here. Bilayer, bi means two, layer obviously means layer, okay? So phospholipid bilayer means we're going to have two layers of phospholipids. And they kind of are reverse of each other. So the tails are next to each other and the heads are outside of the layer. So we're going to learn the heads today are polar, meaning they like water. The tails are going to be nonpolar. And these nonpolar tails and these polar heads, they do a good job of controlling what comes in and out of the cell. And so if you are something that is too large or something that has too much of a charge, you're too polar, you're too nonpolar, they don't let you through because you have to be able to get through the polar section and the nonpolar section. So it's a double, triple, I guess, defense here because you have the polar, nonpolar, then a polar again. So let's write these down. Let's have these on a piece of paper or Google Docs or something like that. So the cell membrane is sometimes called the plasma membrane. It goes back and forth in science. I like the cell membrane. I think it's easy. And so the idea here is cell membrane encloses the cytoplasm. And this is going to be very important for maintaining homeostasis. And if you remember, homeostasis means to keep a balance. And so when you keep a balance, you need to make sure that you have control over what's going in and out of the situation or the system, as we like to call it in science, the system. And so this enclosure around the cell allows us to regulate what comes in the cell and what goes out of the cell. And so the cell has an idea of what can actually come in and out, and we can choose what we need and what we don't need. And especially, we always think about coming into the cell, but also imagine going out. We don't want certain things to go out of our cell, and we want certain things to be able to leave our cell that could be dangerous after they are made in our cell. We call this idea of controlling what goes in and out semi-permeable or selectively permeable. I like to use the word selectively permeable. Semi-permeable, not a big fan. I think it doesn't do a good job of describing the fact that there's a little control here. There's a little control. So semi means in the middle, like there is a little bit. Permeable means letting something in and out. So there's a little bit of letting things in and out. Selectively permeable, the term I like better, means that we are choosing what goes in and out. And I like that better. I think that describes cell membrane better. And so we're going to consist of two layers of phospholipids. We call this a bilayer. Okay, two layers, phospholipids. So phospholipid bilayer is another name for cell membrane. So this phospholipid molecule, we like to call it jellyfish. I hope you like my beautiful drawing here. We have two regions to it. You have the head of it, the head of the jellyfish, and the tails of the jellyfish. Now the head of the jellyfish is polar. It loves water. And we call that water-loving ability of it hydrophilic. Hydro meaning water, philic meaning like you like something. Okay, now this is a term we've seen before. So this always points towards water. Water always, or the head region always points to water, which is always going to be outside the cell or inside the cell. So this is always going to be pointing out of the cell or into the cell, depending on if it's this jellyfish pointing out or this jellyfish pointing in. Next, we have the tail, which is the nonpolar tail. This is made up of a lipid. One is a saturated fat, one is unsaturated. And because it's a mix of both, these kind of phospholipids have a mixture of being something like a solid and a liquid at the same time. We call that a fluid mosaic model. You'll learn about that with your drawing. You had to define it. So it's repelled by water. So anything that's nonpolar is repelled by water. We call that hydrophobic or scared of water. Phobic meaning scared. Hydro meaning water. And so the tails point inward towards each other to form the lipid bilayer. So if we look back here, the tail from this guy is pointing at the tail from this guy. So this is the phospholipid bilayer. Two phosphids, the polar facing out, the tails in the middle. So this is another picture here we got here. We have the phospholipids. We have the lipids actually going in. Okay, and we have the phosphates pointing out. Now, there's other things inside the actual membrane itself, and there's different names for all of them, and we can get really, really advanced with them, and there's some things we will and some things we won't. So there's going to be something called channel proteins. These guys act as passageways, tunnels, and gates to let things through that can't get through the actual phospholipids. So they're kind of like little tunnels. We have receptor proteins that kind of convey an information to the cell or kind of communicate back and forth. So the cells communicate with each other through little chemicals. And so these are little signal receptors that kind of pick up on the chemicals and kind of know what's going on. And then there's marker proteins, which are literally just name tags. Picture just having a name tag on your shirt. It just says, hey, this is who I am. This is what I'm all about. So these three are major things that you'll see on the outside of a cell membrane. If I look here at the picture, you can kind of see we have a receptor protein right here. We have a channel protein right here, kind of lets things get through. And we also have some name tags over here, recognition protein. So you had to make a drawing of that. So hopefully you have all this in your notes. So you had to do the drawing yesterday and the warm-up. Today you did the notes. You wrote all this down. Hopefully you got it all down because I want to see it. This is going to be your spot check for the day. And then you have a homework assignment that you need to do right now. And it's basically over the same exact idea. It is going to require you to draw a picture. So you can do that on Cami, or you can draw it on a piece of paper and turn that in with your assignment. Either way, I need to see your drawing. So just make it happen. Figure out a way. You guys are all problem solvers. Figure it out. So I hope that makes sense. One last thing I did not talk about that is in this picture, and I just keep seeing and I keep ignoring, is the idea of cholesterol. So cholesterol is a steroid. It's 4 rings right here, and it's used as glue. So it's helping glue these phospholipids together. So they could fall apart if the glue wasn't here, but these guys are helping out. And so you see a lot of cholesterol, especially at bends and breaking points of the phospholipid bilayer to kind of help make sure they stick together. So anyways, I hope that kind of makes sense to you guys. If you need any help, let me know, and I'll talk to you guys soon. Happy Thanksgiving. I'll see you guys next week.